today's topic is bacterial recombination, and in that what is conjugation, types of conjugation, plasmids and transposons. So bacterial conjugation, what is conjugation? Conjugation is the genetic recombination in which there is a transfer of DNA from a living donor bacterium to a recipient bacterium, and it often involves sex pilus. There are three conjugative processes, that is F plus F conjugation, and second one HFR conjugation, and third one resistance plasmid conjugation. And we'll see what is individually in the next slides, what is F plus conjugation and what is high frequency conjugation, HFR, which represents high frequency conjugation, and third one resistance plasmid conjugation. So what is F plus conjugation? So genetic recombination in which there is a transfer of an F plus plasmid, that is F factor, from a male donor bacterium to a female recipient bacterium, is called F plus conjugation process, and it doesn't include chromosomal DNA, and which it involves a sex pilus or conjugation pilus. We'll see the process and all. So it involves totally four steps. That is, first one, here we can see the F plus male, which contains F plus plasmid or F plus F factor, fertility factor plasmid, which is a male, which acts as a donor bacterium, and this F plus plasmid contains genes that forms the sex pilus structure, and this serves as a donor bacterium. And here you can see the recipient bacterium, where it doesn't contain this F factor plasmid and it doesn't have the sex pilus structure. So when this, generally what happens, this F plus plasmid undergoes conjugation process and attaches to this when F minus cell. Here you can see the sex pilus adheres to the F minus female cell. Here you can see the F plus donor attaching with the F minus recipient, and in that process F plus plasmid, one of the strands nicks, and thereby that one strand will unwind from the F plus plasmid, and it get traveled along with the sex pilus tube and enter into the recipient bacterium. And here you can see the sex pilus adheres to the F minus bacterium, and one strand of F plus breaks and go through this tube. And finally, after the contraction of this the sex pilus bridge, and which retracts and both the bacteria attaches, and here you can see the one strand of F plus plasmid strand in both the cells, and naturally this one single strand, it forms into the complementary strand. So you will find double stranded F plasmid in both the cells later, as this individual single stranded plasmid, they form its own complementary strand. So now both are double stranded, and here you can see the recipient bacterium is also now become now became F plus cell, as this F factor was delivered to this F minus cell as well. So this is the process of conjugation, where genetic recombination takes place, because sometimes this F plus plasmid will be having this antibiotic resistance gene from the main DNA, and these genes, when they transfer to the F plasmid, and when this F plasmid is transferred to the recipient bacterium, naturally what happens, the antibiotic resistant genes also will be transferred to the another bacterium, and which arises genetic diversity among the bacteria. So this is the conjugation is one of the main process for genetic diversity in case of bacteria. So second one is high frequency conjugation, where you can see the genetic recombination in which fragments of chromosomal DNA from a male donor are transferred to the female recipient bacterium following insertion of F plus plasmid into the nucleus of the donor. So here we will see next slide. So here you can see an F plus plasmid. Uh, Here also F plus plasmid, and in the high frequency conjugation, what happens? Sometimes this F plus plasmid will directly incorporate into the main nucleoid of bacterial, its own bacterial cell. Then this F plus plasmid will be inserted into the main nuclear, and this is called high frequency recombination, because when this when this bacteria, when it attaches to the another bacteria, uh, have the chances that there is high frequency of transfer of this DNA to the another bacteria. So this is called high frequency conjugation. Now here you can see, uh, in case of high frequency recombination, the main nucleoid DNA will one strand will be broken, and the main DNA itself will get transferred to the another bacterial cell. But as you know, the plasmid DNA is very short. It can be easily transferred from one bacteria to another bacteria. But it is the main DNA. It contains thousands of genes. It is not possible for the uh, uh, entire genome to get transferred to the another bacteria, as this conjugation process uh, takes place within few minutes. That's why the entire genome cannot be transferred to the another bacteria. Thereby, there will be loss of much of the genes during this delivery of one strand of DNA to the recipient bacteria. So, in this process, what happens? This the majorly this F plasmid genes may not be completely transferred to the recipient bacteria, and only few genes are transferred there. So yes, you can see that's one single strand, although it is transferring there to the recipient bacteria, but all portion of DNA could not be transferred to the another bacteria, and only few genes will be transferred, and in this it lacks complete F plus plasmid genes. That's why in this case this recipient bacteria cannot have the sex pilus, because here the genome the genes involved for F plasmid are not completely transferred here. Only few genes are transferred, but along with the F plasmid genes, some donor DNA genes, that is which is representing violet color, is transferred here. So, as it is lacking completely pink color here, representing F plasmid genes, it doesn't form the sex pilot. It remains as F minus only. But this donor recipient bacteria will be having some a few extra genes of main nuclear DNA of donor bacterium, and also few genes of F plus plasmid, which finally leads to recombination. And this is the process. And here also similarly with the case of R plasmid resistant plasmid, it also involves, suppose in case some bacteria contains a resistant containing genes in plasmids, and these plasmids, when they are transferred to the recipient bacterium, naturally similar like an active plasmid, resistant plasmid genes are transferred to the recipient bacterium. That bacteria also will become resistant to antibiotics. So these are the various processes involved in the conjugation, and which helps in the genetic diversity in case of bacteria. So in case of plasmids, there are of various types. Here you can see the cell that is, as I already know, it is extra chromosomal DNA, and there are many types of plasmids: conjugative plasmid, non-conjugative, mobilizable, and all. And by function, there are fertility plasmids, which carry genes for pilus and resistance plasmid, and cold plasmid with genes coding for coalescence, and which kill for other bacteria of same species. These four, they are called four plasmids because this was found in E. coli. That's why it's called four plasmids. There are degradative plasmids which are able to digest unusual substances like toluene or sulfuric acid, and virulence plasmid, which makes particular bacterium into a pathogen. And next coming to the episomes. Episomes is a portion of genetic material that can exist independent of the chromosome at some time, while at other times they can be integrated into the chromosome. Examples include insertion sequences and transposons, which are nothing but jumping genes where they can independently exist, or otherwise they can be incorporated into the main genome itself. For example, viruses also and also affected. These are the examples of episomes. The transposons and insertion genes are nothing but the jumping genes. We all know they have the capacity to integrate into the chromosome from inside the output. Factor. Viruses also an example of episome, and their genome can be incorporated into the bacterial genome.